Hi, welcome to Pathology Riddles. Today we will be discussing about a very common infectious disease called malaria. What is malaria? Malaria is a protozoal disease caused by one or combination of Plasmodium species. The species that are most common are Plasmodium vivax and Plasmodium falciparum, followed by Plasmodium malariae and Plasmodium ovale. Plasmodium knowlesi used to infect monkeys has now known to infect humans. Plasmodium knowlesi infection is most prevalent in Malaysia. Have you ever wondered what does malaria literally mean? The word malaria has Italian origin which means bad air. This is because earlier people thought that air over the marshlands causes malaria. Later, Sir Ronald Ross, an army surgeon in Indian Medical Service, found experimentally that female Anopheles mosquito are vectors of malarial parasites. For this discovery, he was awarded Nobel Prize in 1902. An activity for you is to search for Sir Ronald Ross picture on internet. This can be helpful in answering quiz and entrance exams. Let's learn about the pathogenesis of malaria. The definitive host in malaria is the female Anopheles mosquito, where the sexual development takes place, while the intermediate host is human, where asexual development takes place. The mosquito life cycle begins when the female Anopheles mosquito feeds on an infected human. As it feeds on the blood, both sexual and asexual form of parasites enter into the mosquito. The asexual form dies, while the sexual form or gametocytes further develop. Once it reaches the stomach, exflagellation takes place. That is, 8 thread-like structures called microgametes are formed from one microgametocyte, while one macrogamete is formed from one macrogametocyte. Fertilization occurs when one microgamete penetrates one macrogamete. Fertilized macrogamete is called a zygote. Zygote lengthens and matures into ookinete, which is motile. It penetrates the stomach epithelial lining of mosquito and lies between the peritrophic membrane and epithelial cells. It then develops into oocyst. Inside the oocyst, there are hundreds to thousands of sporozoites which develop. After around 10 days, oocytes mature and rupture, releasing sporozoites. These sporozoites are released in the body cavity of mosquito and have special liking towards the salivary gland. So when the mosquito bites a healthy human, it transfers these parasites from their salivary gland duct into the bloods of the human. The human life cycle mainly have zygony and gametogony phases in the following order. First is pre-erythrocytic zygony or primary exoerythrocytic zygony. Second is erythrocytic zygony. Third is gametogony. In Plasmodium vivax and Plasmodium ovale species infection, there is another phase called secondary exoerythrocytic zygony. Starting with the zygony phase, pre-erythrocytic zygony. This occurs before the erythrocytic phase, hence called pre. As the sporozoites enter into the human blood, it reaches into the liver parenchymal cells. There it further develops into pre-erythrocytic zygont, which when the development is complete, the liver cells rupture and the zygont release thousands of merozoites into the bloodstream. The duration of pre-erythrocytic phase varies according to the species. Example, 6 days in Plasmodium falciparum, 8 days in Plasmodium vivax, 9 days in Plasmodium ovale, and 13 to 16 days in Plasmodium malariae. Next phase is erythrocytic zygony phase. In this, the merozoites infect into the RBCs. They develop into trophozoites, then scisons, and release merozoites. Following erythrocytic scisony occurs gametogony. After few cycles of erythrocytic scisony, some merozoites in the RBCs develop into male and female gametocytes known as microgametocytes and macrogametocytes respectively. The cycle continues when a female Anopheles mosquito feeds on an infected human. Let's discuss secondary exoerythrocytic scisony phase, also called dormant scisony. Some sporozoites, on entering into the liver, enter into a dormant stage where they don't undergo multiplication for some time, for weeks, months, or up to 2 years even. They are called hypnozoites. Later, when they are reactivated, they become secondary exoerythrocytic scisons and release merozoites, which again infect RBCs. This is called relapse of malaria, which occurs mostly in Plasmodium vivax and Plasmodium ovale. Let's now discuss the clinical features of malaria. The patient usually presents to emergency with febrile paroxysm. What does this mean? The patient experiences 3 successive stages beginning from early afternoon. The first stage is cold stage, where the patient experiences intense cold and shivering. It lasts for around 15 to 20 minutes. Following this, the patient experiences hot stage, where the patient has high fever, headache, nausea, and vomiting. This lasts for around 2 to 6 hours. The third stage is profuse sweating. This febrile paroxysm occurs every 48 hours in Plasmodium vivax and Plasmodium ovale, where the fever is called benign tertian. The fever occurs every 72 hours in Plasmodium malariae, hence called quartan fever. In Plasmodium falciparum, the fever is called malignant tertian. But why? The febrile paroxysm corresponds to the completion of erythrocytic scisony. During this phase, the scisoin ruptures, releasing merozoites, malarial pigments, and debris. In Plasmodium vivax, ovale, and malariae, the cycles of different broods of parasites synchronize and the scisons rupture together. While in Plasmodium falciparum, the cycles of different broods of parasite do not synchronize, hence they rupture at different times. Hence there is fever every 24 hours. It could also mean that there is a mixed infection. That is, along with Plasmodium falciparum, one more species of malaria has infected the same patient. The second clinical feature is anemia. Anemia may develop after few paroxysms. This is due to RBC lysis, that is due to splenic removal of infected RBCs or uninfected RBCs with immune complexes, where antibodies bind to the antigen of malarial parasite. The third clinical feature is splenomegaly. This is due to proliferation of macrophages. These macrophages are involved in phagocytizing the parasitized and non-parasitized RBCs. We also need to know about pernicious malaria. It is a complex of life-threatening complications seen in some cases of acute falciparum malaria. It is of 3 types. Cerebral malaria, characterized by hyperpyrexia, coma, and paralysis. Algid malaria, characterized by shock. Septicemic malaria, characterized by high fever and involvement of various organs. These complications occur due to heavy parasitic load. Another complication of Plasmodium falciparum infection is black water fever. When a patient with falciparum infection is inadequately treated with quinine, antibodies are formed against this quinized RBCs. When the patient is infected again with Plasmodium falciparum, the administration of quinine results in autoimmune mechanism. There is massive destruction of RBCs, fever, hemoglobinuria, and renal failure. We have come to the end of this video. In the next video, we will be discussing about the laboratory diagnosis of malaria. If you like the video, please like and subscribe. Till we meet the next time, this is Dr. Bismay signing out.